Anu, shall we go ahead?
Looks like people are still joining, so maybe let's wait one more minute.
Okay.
Hello.
Hi, Stefan.
Glad to see you, Ian.
Same here. I always love your hellos. It always makes me smile.
All right, with that hello, I think we'll get started, Chen.
Great. Well, thank you so much, Anu. And yeah, my name is Xinwei Wang, and I am the co-director for the CCI Liver Cancer Program. And today is our very honored to introduce our today guest speakers of this month by Dr. Hien Dang. I'm going to say a few words about the Hien. Hien received bachelor degrees in bioinformatics from Michigan Technological University and then PhD in molecular medicine from Penn State College of Medicine. She went on to do a postdoc at the NCI, then moved back into Philadelphia to establish her lab, now accounting about over 8 years ago at Thomas Jefferson University. Now, currently, she's a vice chair of the research and chief of the Division of Surgical Research at the Department of Surgery, and also assistant professor at the Sidney Kimmel Cancer Center of the Thomas Jefferson University. Now, her lab is really focusing study on oncogenic role of RNA binding protein in liver cancer, as well as pancreatic cancer, I assume, using both genomics, bioinformatics, translational science, and molecular biology, particularly utilize CRISPR-Cas9 technology to study the function as well as the RNA biology. Now, in addition to bench science, her team is currently also collaborating with many medical oncologists and surgeons to develop a biomarker to identify patients at high risk for liver cancer. She oversees both liver and pancreatic cancer biobank with many translational projects at the Kimmel Cancer Centers, which I heard that recently just to be converted to a comprehensive cancer center. Congratulations again. Now, in addition to her scientific work, she has been quite active in leadership role, including her initial tenure at the NIH. She served as a multiple committee, including NIH Fellows Young Investigator Committee called FUNCO, and Asian Pacific Islander American Association, as well as RNA biology interesting group. Now, among her many accolades, and I was counting, and some of those noticeable include the NCI Director Innovation Award, NIH Asian Pacific Islander Young Investigator Award, and NCI Director Award for Merit for her scientific work, as well as receive numerous awards, including the American Liver Foundation Charles Trey MD Memorial Liver Scholar Award, among many others. So the most notable, I would say is she's currently a Cancer Moonshot Scholar, which is supported by the White House to advance cancer research. This is really not only quite a big honor, but also huge monies that go into her lab for the study. This is absolutely had to be congratulated for receiving such a good recognition. The title of her presentation is, I really quite like it. It's called "How Small Change Can Drive Cancer Progression." This is quite interesting. Without further ado, Hien, the stage is yours.
Sure. Wow, that was fantastic. You make me sound so much more impressive than I really am. Thank you for the introduction. My lab focuses on both liver and pancreas, although I came from a liver background. Pancreas is something that we've just started doing in the last couple of years, as I started taking on more administrative roles. I'll start with introducing hepatocellular carcinoma. Majority of the talk will be focused on HCC, and towards the end, there's a little bit of the research program that we've just started building. and in pancreatic cancer. So I'm not going to spend too much time talking about HCC in this group, because I know everybody appreciates how much how important HCC is, and how we should be spending more time studying it and trying to figure out different molecular mechanisms that can really help us develop better therapies. But what I will really want to highlight is that HCC is a very heterogeneous disease, and because of this heterogeneity, it is quite a burden towards our HCC cancer patients, because it makes it really difficult tumor to treat. And HCC is, because of the heterogeneity of the tumor, it's mostly attributed, this heterogeneity is mostly attributed partly by cellular plasticity, a mechanism that enables cells to acquire new phenotypic and functional features by transitioning across distinct cell types that contribute to tumor, that has been demonstrated to contribute to tumor initiation, progression, metastasis, as well as resistance to the certain therapies. And this cellular plasticity is an emerging hallmark of cancer that was recently described by Hahnemann in Cancer Discovery in 2024 paper. Dr. Robert Weinberg was not a part of that piece, although I'm pretty sure he contributed tremendously. So its cellular plasticity is often governed by epigenetic and transcriptional reprogramming, and has been linked to the microenvironment, often responding to a specific stimuli or stress response. A great example of cellular plasticity is EMT, or epithelial to mesenchymal transition. While cells with an epithelial phenotype can transition into a mesenchymal-like phenotype while maintaining the capacity to stay within the epithelial state. And this allows the cells to essentially migrate and invade. And then you have the mesenchymal to epithelial transition once it's in the distant organ systems, where then it can, again, change its cellular state so that it can colonize and form tumors. This process on its own can lead to dynamic changes in gene expression and a lot of structural change within the chromatin. And what our lab is essentially trying to ask and really focus on is identifying true mechanisms or drivers of this phenotype that not necessarily is driving the changes, but more or less regulating these changes or mechanisms that promote these dynamic transcriptional programming. And one group of proteins that we think are really important in maintaining this is RNA binding proteins, as they are regulators of the transcriptome. And there's multiple different RNA species, and these different types of RNA binding proteins can modulate cellular homeostasis or this reprogramming really well. And that's not something that's conceptually novel, in the sense that these proteins are master regulators of the transcriptome and have been implicated in cellular plasticity before, specifically looking at EMT. So there have been proteins such as the Musashi protein or the HUR RNA binding proteins that have been demonstrated before to be either promoters or suppressors of EMT. So there are 1,500 known RNA binding proteins, and they are really important in all aspects of cellular homeostasis, regulating RNA biogenesis, transcription, to repression of RNAs, as well as translation of different RNAs. And RNA binding proteins tend to form biomolecular condensates or membraneless organelles composed of concentrated biomolecules, including proteins and nucleic acids. These organelles are dynamic and can be important in diverse processes, including RNA metabolism, ribosome biogenesis. They can react to DNA on damage response, as well as signal transduction. And they are mostly found in the nucleus, as you see here. They're highly involved in transcription, whole heterochromatin, DNA replication. And these essentially are really important for maintaining cellular homeostasis. However, they do also exist in the cytoplasm. More well-known are stress granules that then form these condensates under stress conditions, and these stress granules have been implicated to be really important for tumor genesis as well, and this is specific to pancreatic cancer. In order for these RNA binding proteins to interact with other RNA binding proteins or nucleic acids, they undergo a process known as liquid-liquid phase separation. And this LLPS is driven by multivalent macromolecule interactions. That's important for biomolecular condensates. This is a process where RNAs and RNA binding proteins can interact and dynamically organize itself into these membrane-less organelles, and that this organization can really facilitate signaling or cellular organization within the cell itself. This whole process requires an intrinsic disordered region or a low complexity sequence. And these low complexity sequences are found in more than 95% of all RNA binding proteins. So these regions are really important for the protein itself to really undergo liquid-liquid phase separation, and then its ability to interact with nucleic acids or other RNA binding proteins to form these condensates. So if you want to imagine liquid-liquid phase separation is essentially the same concept of oil droplet in water. There are two distinct phases that never really mix. Even if you just take the water and the oil and you shake it quite hard, you think that it's mixing. It's actually really not. What it's undergoing is these tiny little droplets that you don't really see. And then if you just let it settle over time, it eventually comes back to these large distinct droplets. And so it looks kind of like this. So they never really truly mix, and they're distinct organelles or distinct phases within a solution. And so why do we think that RNA binding proteins are important, and why do we think that they are important in regulating cellular plasticity, and why do we think we need to look at it? Well, in general, RNA binding proteins are upregulated across multiple different cancers. And we've shown this in our own cohort as well in 2017. There's more RNA binding proteins that's overexpressed in cancers than there are transcription factors or other protein coding genes. And when we look at RNA bindings as something that's important for tumor progression, we see that RNA binding proteins in general are more likely to be selected over time or during tumor evolution. So if you look at the integration between RNA and gene copy number, we can see that there seems to be more RNA binding proteins that have increased gene copy number, but also have an increased mRNA expression. And so this suggests that RNA binding proteins, or the deregulation of RNA binding proteins, are selected during tumor evolution. What's really interesting is that although RNA binding proteins are more likely to be altered in cancers, and that they seem to be more selected during tumor evolution, their changes are really, really subtle. There's only a few RNA binding proteins whose gene expression is more than twofold. In fact, in our recent cohort, what we found was that when you're comparing RNA binding protein expression from tumor versus adjacent non-tumor, we found that the fold chain of RNA binding proteins at most is about 1.5-fold change. And so these changes are really subtle. So what we're interested in is asking is, if these RNA binding proteins are so important, they seem to be selected during tumor evolution, why are the changes that we're observing so little and so subtle? And so the next few couple projects is going to be focusing on one RNA binding protein, negative elongation factor E. And this essentially tries to tease out the reasons why one small change can really have this big impact, and whether this impact can really drive tumor genesis. And so I'm not sure if we're going to get there, but I'm hoping that I have enough information or data to allow you to believe that this is something that is conceptually interesting and does really occur, and that RNA binding proteins are that group of proteins that can get us there. So, of course, lab is not really a lab unless we have little minions working on them, and I have probably the best team. I don't know how I've been so lucky since I started my lab 5 years ago, but the work that I'm going to present is strictly done by Anna, Alvaro, and Kay. And like I said before, I don't know how lucky I've been, but I've got probably the best lab and the best teammates, and they've really made this project really possible. And so these are our faces. And if anyone's interested in looking for a research tech around the Indiana region, Anna is leaving, and she is phenomenal. So she is looking. So we start with a gene candidate approach, and we literally just focused on one of the most highest genes, the RNA binding protein with the highest expression in HCC. And we did this by just looking at gene copy number alteration between RNA and gene copy number. And we found that NAFLD is among the highest between two different independent cohorts. And NAFLD is a part of the negative elongation factor complex. And this protein is involved in binding nascent RNAs during transcription. And then this interaction with the nascent RNAs is what allows this polymerase to pause. And RNA polymerase pausing is a mechanism that allows transcriptional pausing to occur. So pausing is essentially a mechanism that allows the cells to actively transcribe genes at a very fast, efficient way. And so if stress is occurring, then the chromatin's already open. And so then essentially what happens is the NAF complex gets released, and then elongation can occur really fast. And so NAFLD is a part of this complex. However, there's been many studies out there that's demonstrated that NAFLD does, in fact, function independent of this NAF complex as well. It's also really important for us, and it has been demonstrated to also undergo phase separation or liquid-liquid phase. However, it's been demonstrated to undergo liquid-liquid phase separation as a complex. This is not something that's really novel. And so based on what we have read before and what we thought, we wanted to focus on negative elongation factor E because one, it has a really low, the change in gene expression between versus non-tumor was not very high. It seemed to be really important in the sense that it was selected during tumor evolution. And then it has somewhat of an RNA binding protein characteristic, but it has never really been demonstrated to be an RNA binding protein. So we sought out to really first test whether NAFLD is really important for HCC progression. And when we looked at NAFLD expression over tumor progression, we saw that its expression level in cystic nodules are pretty low. And that as the progression increases, so you have here low grade to high grade, and then early onset HCC, low grade dysplastic nodules to high grade dysplastic nodules, you see that NAFLD expression slowly increased up to early HCC. So there seems to be a really nice progression of this expression of RNA binding proteins, but then it really just kind of plateaus out. And then we found that when we knocked down NAFLD, both in vitro, and I'm only showing you the in vivo here for the sake of time, that they actually, tumors just do not initiate over time. And this is done in one HCC cell model here in the HGH1. And then we also show that NAFLD is really important in MYC-induced HCC models. We found that when you knock down NAFLD, MYC signaling is significantly reduced. And so we went to the animal, and this is a Sleeping Beauty MYC model. And when this was done with Wen Shu at UMass. And so what he did was overexpress the human form of NAFLD in his mouse P53 knockout mouse models. And he found that NAFLD actually really didn't do much. When you look at the actual liver, there was really no phenotypic effect. There was nothing wrong from just looking at the liver. It was really still very nice and well colored. But when you overexpress MYC, we already knew this, MYC by itself can really promote tumor genesis in the P53 knockout system. But when you overexpress both MYC and NAFLD, you were able to double the number of tumor nodules. So this suggests that NAFLD is really important for tumor genesis, but not necessarily by itself. It requires a second hit. And so one of the things that we wanted to really focus on was whether NAFLD is really a bona fide RNA binding protein, because it's yet to be demonstrated as such. And so we did confocal microscopy, and what we found was that these NAFLD was forming these really nice foci. So there are tiny little dots here, and NAFLD is in red. And we asked whether these foci or NAFLD bodies were co-localizing with other nuclear bodies that are membraneless organelles. And we found that, in fact, actually NAFLD was really by itself. It never really did co-localize with any of these other nuclear bodies. And so that led us to think maybe the RNA binding protein NAFLD is potentially important by itself, and it can undergo liquid-liquid phase in our cancer cell models. And so we collaborated with Nicholas Fauzi at Brown University, who's a biophysical biologist, who actually trained at the NIH, and we overlapped a little bit. And and this is completely outside our field, and he looked at the protein and the structure of it, and he used this program called Disordered, and he found that within the NAFLD sequence, there were 2 low complexity regions, and these regions had a really high disorder prediction. And so this is essentially a way for us to know whether the protein itself can undergo liquid-liquid phase, because these regions here are also known to be very important for liquid-liquid phase separation. And so what we did was clone NAFLD full length and then generate the different mutants, and then also clone the RNA recognition motif, which is required for interaction with single-stranded RNAs, and sent it to Nick at Brown. And a few months later, he came back and he showed us that with the addition of RNA, NAFLD with RNA was able to form these beautiful liquid droplets in a test tube. But when you remove the low complexity sequence here, you see that they were able to form these droplets, but they're a lot smaller and there seems to be more of them. But when you remove the LCR2, which is a huge chunk, it's almost 60 or so amino acids, you lose this ability completely. And what's really interesting, and we thought the RNA recognition motif might be really important. In fact, we didn't really see much of an effect. When you remove the RM, they were able to still form these really big, beautiful droplets, although not as many, but they were still able to. So that was really interesting and surprising for us. And this was one of the things that we thought we must be onto something, and that this is indeed a true RNA binding protein, because recently people have really shown that NAFLD by itself does not form phase separation or does not form these liquid droplets. It requires the whole complex. So what we did was then generated a CRISPR knock-in system where we can really follow the NAFLD and really test to see if NAFLD can really undergo liquid-liquid phase separation in the cells. Majority of the literature uses an overexpression backbone, and there's a huge limitation with that, just because an overexpression of NAFLD in any cell type can cause cell death if it's overexpressed long enough. And so we decided that we had to use the system, even though it took us a little bit of time to get there. So essentially what we did is just cloned a donor arm with about 800 base pair homology arms near the start site or the stop codon, and then had a guide RNA cut where right before the stop codon, and then essentially transfected 2 plasmids together to generate this knocked-in loci where NAFLD, where GFP is tagged right before the stop codon of NAFLD. We were able to successfully do this in 2 different HCC cell line, one where we had a complete knock-in using TA cloning. We were able to see that every single allele had this GFP. And then in the hep3b cell line, we were able to only do this in about, they had about 3 alleles, so only 2 of those alleles were able to be tagged with the GFP, whereas the other allele was not, as you see here. But phenotypically, when we're looking at under confocal microscopy, we're able to see some really beautiful foci, as seen here in the hep3b and in the HLE cell lines. So this is really beautiful. Like now we have a beautiful system where we can really look at the foci and really test the hypothesis that NAFLD can undergo liquid-liquid phase separation. And so one of the first things to show that NAFLD can undergo liquid-liquid phase separation is by performing fluorescent recovery after photobleaching, or FRAP. And this is a method that essentially you draw a really nice ROI, and then you shoot a 405 laser to bleach cells at 100%. And we've gotten pretty good. It took us a little bit of time to really just wrap these tiny little foci without having to affect the entire cells, because that can happen, and that bleaching can really have an effect on the cell. And so you're more likely to see background noise than a true signal. But as you see here, we were able to kind of frap here. This is the foci. Here's the bleach where we frapped it at 100%. And within 1 second, we started to see it come back. by 2 seconds, you're already starting to see it really come back. And by 3 and 5 seconds, it's pretty much back to where it was originally, although not as bright. And so this is the litmus test to really test whether your protein or any RNA binding protein can undergo liquid-liquid phase separation. So why is this important? Because if this is possible, that means that these NAFLD foci are potentially biomolecular condensates that can phase separate to then interact with multiple different various proteins or nucleic acids that can then potentially have some type of effect on signal transduction. Another litmus test to determine whether this protein or these NAFLD bodies are molecular condensates is by treating them with 1,6-hexanediol, an alcohol that blocks weak interactions required for the biomolecular condensates to form. And so what we did was essentially treat it, took pictures, and it's not very quantitative, it's very qualitative. It's one of those things that you kind of just look at by eye. If it's there, it's there. If it's not, it's not. And so when we treated it with 10% hexanediol, at 5 minutes, we started to see that some of these foci start to really disappear. So we're essentially breaking that interaction. So that's why you don't see them anymore. And this is also evident in the other cell model that we generated. So together, what we found was that these NAFLD bodies do exist and that these NAFLD bodies can undergo some type of dynamic change or phase separation. And so we wanted to next test whether these LCR regions are really, truly important for this phase separation mechanism. And so we did an overexpression model. We really had to titer and use very, very low MOI to get this phenotype. Because again, what I mentioned was that when you have too much of this, the overexpression of NAFLD, the cells tend to die. And what we also observed was that when you have such a high level of NAFLD, instead of forming these beautiful foci, we start getting sheets. And so this was the limitation in an overexpression model that we were observing. And so that would explain why other labs were not able to really say that this NAFLD by itself can undergo liquid-liquid phase separation, or they're in fact phase separating into these beautiful foci. So if you're using an overexpression model, you're more likely to get these sheets. Although we did try to frap the sheets and they did recover, we just weren't sure if that's something that's real or just clunks of things. But it was worth noting that we did try. So what we did next was test whether these the deletion or the mutants actually had an effect on cancer phenotypes. So we looked at cancer cell proliferation. We also looked at colony formation. We're currently looking at migration and invasion as well. But so far what we found was that if you have a mutant of the DLCS1, DLCS2, when you inhibit the ability of the cells to undergo liquid-liquid phase separation, you cannot rescue cell proliferation or colony formation. And we've repeated this in also the other cell model, but I'm just only showing you the Hep3B here. And so what we think is happening is that this LCS1 and LCS2 is really important for liquid-liquid phase separation and potentially downstream signaling. And so we wanted to now generate a knock-in system where the LCS1 and the LCS2 is deleted. And we weren't going to use an overexpression because of the sheets that we were seeing, which is not physiological anymore. So the best way was essentially to utilize CRISPR to then generate these double cuts. So what we did was cut the beginning and the end of the LCS1 region, and we were lucky enough to be able to generate these lines because, God forbid, it took us a really long time to get here. And here's the Western blot essentially showing you that we were able to successfully cut these genomic regions, and the protein were not awfully affected, and we were able to still see that NAFLD is still expressed. And you see 2 bands here because again, Hep3B cell lines only about 2 of those alleles were tagged. The third one was not tagged, so you would expect to see a wild type. But the cut, the LCS cut that we saw is a complete cut. And then in the HLE cells, we were able to do that completely without even worrying about an endogenous having an effect. And so what you see here is that when you have this LCS1 deletion, there seems to be more foci and they seem to be smaller. This is a lot more evident in the HLE cell lines you see here. So we went out to ask if this is something that we can quantify. So we worked with our core facility here in imaging, Jason Hill, and he was able to look at each single foci and really analyze for us. We asked, are there more foci when you have this mutant? And if there are more foci, are they smaller or bigger? And in both cell lines, what we saw was this beautiful trend that in general, you were seeing more foci, and these foci were generally smaller. And so we have this really beautiful phenotype. When you remove the LCS1, you started to see phase separation being a little bit affected. And that we also saw was that you get more. And this is very consistent with what we were seeing in vitro test tube at Brown University. So we opted to go and try and frap these little foci. And indeed, we were able to frap them. And what was really interesting and what we expected was that when you delete these regions, you should expect to see an effect on phase separation or its dynamic ability to recover. In fact, we actually didn't see much of a difference. And this has been repeated. So what you're seeing here is about 15 cells, and it's one single foci per cell. We've done it where we've frapped multiple foci per cell as well, just to make sure that we're seeing the same phenotype. We've extended this to 5 minutes to make sure that the signal that we're seeing remains stable, and it does remain stable. And so this is surprising in the sense that we were expecting a much more dramatic effect in the dynamic, but we're not. But we're still seeing more. And so what we're thinking is that maybe these the LCS1 region is not necessarily important for inhibiting or completely affecting phase separation, but maybe it's just affecting what it's phase separating with. And so I think I went too far. Let me go back. And so based on this, we wanted to next ask whether this LCS1 or the LCS2 is important for any type of phenotypic changes that might be important for, genotypic changes that might be important for cellular plasticity. And so we started looking at chromatin accessibility. And the reason why we started there is because previous evidence in our lab, as well as others, has shown that NAFLD is really important for maintaining chromatin accessibility. In our lab, we were able to really just show that when you lose NAFLD, MYC binding to specific targets is significantly reduced, suggesting that NAFLD is really important for some type of chromatin accessibility for MYC interaction. And so we sought to ask, what happens when you delete NAFLD completely to chromatin accessibility? And what we found, and when we performed ATAC-seq for this, and what we found was that when you remove NAFLD, the chromatin is less accessible. And when we overexpress NAFLD, we're able to rescue and really amplify that signal. And this, again, is really showing that if you have too much of NAFLD, there's a lot of background that can happen. And so we also perform H3K27AC chip. And the reason why we did this was to show that these regions are actively transcribed. And so what we're saying here is that when you remove NAFLD, chromatin accessibility is lost, and these are regions that are actively being transcribed. And the majority of these regions are found within the promoter regions, where some in the intergenic. So our next question was essentially, well, if you've lost NAFLD, and NAFLD is important for chromatin accessibility, what happens to MYC? And the reason why we focus on MYC is because we've, again, shown previously, MYC is really important for NAFLD-induced HCC, because it is with MYC that NAFLD really enhances the signaling to promote HCC progression. And so we wanted to know if what we were seeing in our previous work, that when you lose NAFLD, MYC activity for a certain, MYC is not bound to a few genes, but whether this is something that's MYC generalized. And so what we did was perform MYC chip, and then looked at the ATAC-C regions, so the chromatin accessible regions. And we performed this in our CRISPR lines where NAFLD is completely lost. And what we found was that when MYC was able to bind in the control, but when we removed NAFLD, a lot of the MYC regions were significantly lost. So MYC was no longer binding in these regions. And we think it's because these regions are no longer accessible, and that's why. And so this data combined tells us that NAFLD is modulating chromatin accessibility, and this is having an effect on MYC's ability to come in and bind to the consensus peaks. And then, of course, we wanted to know if this is through liquid-liquid phase separation. So we asked and did the same experiment again with ATAC-seq. We've done also MIC chip as well, but the data hasn't come in yet. And this is data that we just generated yesterday, practically. And what we found was that when you knock down NAFLD and rescue with full length, delete the LCS1 or LCS2, and you looked at only the MIC regions, what we found was that NAFLD full length was able to rescue these MIC regions, but the LCS1 and LCS2 compared to full length was not able to fully rescue. So this suggests that the LCS1 or liquid-liquid phase separation of NAFLD is really important for MIC binding to the chromatin regions, possibly through chromatin accessibility. So we wanted to know next if this is something that is through NAFLD's interaction with the chromatin. And we did NAFLD chip, and we found actually there's a lot of background noise. So NAFLD not necessarily was binding to the chromatin to have this effect. And it's well established that NAFLD is not really something that really binds to the chromatin itself. It's a part of the complex, but it's never really been shown to really interact with the chromatin. And even when we perform our own chip assays, we were not really seeing a lot of really strong signals either. So if it's not binding to the chromatin, what is it doing? Or how is it like modulating the chromatin? And so what we did was a really quick and dirty experiment. We performed an IP and then sent that IP lysate for mass spec. And although it's quick and dirty, we also really focus on cytoplasmic versus nuclear. And we only focus on anything that was nuclear because we wanted to know whether these nuclear or the foci is what's affecting it, not just NAFLD in general. We did this in 2 different HCC cell lines as well, just to make sure that what we're seeing is not a cellular or cell line artifact. This is something that might be potentially important. And what we found was about 136 proteins. And of those, 2 chromatin modulators showed up, and SMARCB1 was one of them. And we chose SMARCB1 to work with it because there's previous evidence that shows that SMARCB1 interacts with MIC to modulate MIC interactions or MIC signaling downstream. And so our question was, does NAFLD modulate the chromatin through SMARCB1 to enhance MIC signaling? And what's really interesting about SMARCB1 is that it's actually been shown to be a tumor suppressor when it interacts with MIC. This is well established in a couple of different cancers, including rhabdoid cancers. And what they show is that MIC interacts with SMARCB1, which is also known as SNF5, and actually represses MIC activity. And this repression is usually lost because MIC or SNF5 is usually mutated in these cancers. And so it causes a loss of function. And so MIC then is gone unchecked. And so now you have oncogenic activation in their cancer. But what's really interesting in HCC was that there's a 2021 paper that showed that actually SMARCB1 is highly expressed. It's not at all deleted. And this is evident in 2 different cohorts, including the TCGA as well as ICGC. They also show that SMARCB1 expression is increased across or throughout tumor progression as well. And so we have this contradictory concept, but what we think is happening is that SMARCB1 is probably not repressing MIC, and that SMARCB1 and MIC is really important, and that NAFLD is probably what's driving this interaction. So our hypothesis is that SMARCB1, that NAFLD is important for SMARCB1 and MIC interaction for this oncogenic phenotype that we're seeing, at least in HCC. So to first verify our proteomics data, we performed IP and found that indeed SMARCB1 does interact with NAFLD. It also does interact with MIC, and that MIC does somewhat interact with SMARCB1 as well. And so we've done this in 3 different cell lines. I'm only showing you again in hep3B just for the sake of time. And then we also look to see if NAFLD is really important for SMARCB and MIC interaction, because our hypothesis is that NAFLD is required for this interaction. And so we can see here that when we knock down or lose NAFLD, we see that the interaction between SMARCB1, this is a SMARCB1 IP, you see that there's a loss interaction between MIC and SMARCB1, but you're able to kind of rescue that here when you overexpress NAFLD in both HCC cell lines here. And so the next question for us is whether the LCR1 or the LCR2 is really important, or phase separation is important for this interaction. And so again, we did an IP analysis, and where we showed here with the full length, you're able to see that the interaction between SMARCB1 and NAFLD and MYC is there. But what happens is that if you have the LCR loss, that interaction with SMARCB1 and NAFLD is still there, but mixed interaction with SMARCB1 is lost. So this is telling us at least that the LCR1 or liquid-liquid phase separation is really important for the SMARCB1 MYC interaction. And maybe the NAFLD is modulating chromatin accessibility through these phase condensates to allow more of accessible chromatin that then allow the recruitment of MYC to then bind to its regions and then promote downstream transcription. And so this is our model of liquid-liquid phase separation. And again, we were saying is that HCC and these biomolecular condensates consist of nucleic acids as well as different RNA binding proteins. And that when you have high NAFLD levels that we see in HCC cell lines, they seem to like undergo more of a phase separation. There seems to be enhanced because phase separation is highly driven by concentration as well as pH. In our case, we think NAFLD is acting as a scaffold, and that this increase or extra expression of NAFLD is causing more interactions or enhanced interactions or more biomolecular condensates that then can recruit various proteins, including MYC, to then have more of an active downstream signaling and promote cancer. And so this is like something that we're still trying to kind of put together, and we're hoping to submit this in the next 2 or 3 months. A lot of the data is being done or processed right now, and we're doing the in vivo, the xenograft models to show that the LCR regions are important for tumor initiation. We're also looking at doing the IP to show that with the deletion of the LCR2, you would also lose the mark and SMARCB1 and the MYC interaction. And so this project is hopefully going to get submitted in the next 2 months and published somewhere. So I'm going to switch gear a little bit because, you know, our lab is kind of diverse and it's gotten to be quite complicated in the sense that my role as a vice chair of research in Department of Surgery. The other aspect of our projects is looking at how we can then target NAFLD in more of a translational project. And this project is mostly driven by Laura Reynolds. She's a brilliant graduate student in our lab. It really took on this project headfirst with really no experience at all, but she's done a great job at generating a lot of this data. So this was kind of an accidental project where when I was a postdoc, we were looking for genetic mutations that could potentially explain why we're seeing what we're seeing in our HCC models. And what we found was that there was no mutations. We did a lot of work and a lot of sequencing, but there was no mutations. But we serendipitously designed primers within the exon or intron regions and found that there was indeed a SNP or a single nucleotide polymorphism within these HCC patients. And we sequenced both tumor and non-tumor. And so this is a germline SNP that we found. It's a G to A mutation, and it's only found on one allele. And what's really interesting was that although NAFLD is amplified, and that could contribute to its increased gene expression, we found that when you have with patients with HCC that have the SNP, they actually tend to do better. They also have a much better disease-free progression curve. And so this was interesting to us because we're wondering, well, if NAFLD is oncogenic in HCC and its high expression is important for driving this MYC-induced HCC phenotype, why would you have a SNP that can actually make you do better? And so we did some analysis where we looked at the SNP by itself, and we found that 5% of the SNP is in Southeast Asians, but in general, in the general population, the SNP is only found in 1%. Because our cohort is majority HCC from HBV patients, we found that the SNP is highly prevalent in HBV HCCs. And that, again, if you did the analysis in terms of just looking at months of survival, we saw that patients with the SNP had about 21 more months survival than the wild type patients. And that is also seen in recurrence or progression-free survival. So we had a postdoc from Wuhan University come, and he came from a splicing lab. And right away, because this was literally one of those projects where we put in the back burner, because we didn't think much of it, because the number of patients that we found the SNP in was like 7, and the patients that didn't have the SNP was like 90. And so it was one of those things like, is this real? And so what we did was, what he did was said, well, maybe it's important. And so he found that the SNP was found near the polyperimeter tract, which is where spliceosomes spined. And so he had this hypothesis that the SNP actually has an effect on splicing. And so he generated a mini gene with GFP, and then he overexpressed it in HCC cell lines. And design primers were at the GFP and then exon 9. And the reason he did that was he wanted to make sure that we're only capturing the exogenous SNP that we're seeing, not anything endogenous. And what he saw was that when you have the SNP, there was a lower expression of NAFLD. And when we put this into a luciferase backbone so that we can control for transfection efficiency or concentration of transfection, we found that, again, if you have the SNP, the luciferase signal was really, really reduced. And so this led us to go back to our HCC samples and look at the RNA expression of NAFLD between patients that had the SNP and the wild type. And what we found was that patients that have the SNP tend to have a lower NAFLD expression. What's really interesting is that it didn't matter. NAFLD was just high in tumors, but if you had the SNP, your NAFLD levels were a little bit lower. Although this is significant, the N of this group is really small, and we're really working on this. And Laura has expanded on recruiting patients that have both HCC and PDAC to see if we can identify the SNP. And so far, we have not had much luck because the majority of the patients that come to TJ are not Asians. And so we're working on translating the IRBs or translating the consent forms to various different languages so that we can capture all the patients. So what we've done so far is to capture almost about 400 patients, and we've still only seen 8 patients with the SNP. So I think this is going to be one of those really massive studies that might not go very far. And the rate limiting step is that only 1% of the patient population or 1% of the general population has the SNP. And so what we're trying to do is really capture more patients or Asian patients because they have a tendency to have a higher percentage of the SNP. And so another way to really study splicing is by developing antisense oligonucleotides, or ASOs, and they essentially are designed near the exon-exon junctions, the 5' and the 3'. And when they're added to the cells, essentially what they do is interact with the pre-mRNA, bind to it, and block splicing, and that can then cause a splicing event. And in our case, what we found was that in addition to the decreased level of NAFLD expression, we found that the SNP itself caused an exon 10 exclusion. Because when we did the mini gene assay as well, we found that there is a lower 103 base pair loss in when we sequenced it. And so the SNP was causing this exon 10 exclusion. And so we developed these ASOs to target exon 10. And here, you can see that we've done this in pancreatic cancer cells, and we've done this in 5 different HCC cell lines and 3 or 4 different pancreatic cancer cell lines. And we still always see the same thing, that when you add these ASOs to these cells, you see a significant reduction in the wild type NAFLD or the full length NAFLD, but you also see an increase in this exon 10 isoform. We also see that there's a change in protein, but we're not very sure what this really means. It could be a byproduct of something else, or it could be that the SNP is producing a protein. So we're currently investigating this and really trying to figure out if this is something that's real, or it's a byproduct of the ASO itself, or it's probably just a protein thing from the antibody blotting. So we're really looking at this right now and using these ASOs. But our lab is mostly interested in using these ASOs to really target tumor burden, as well as these molecular condensates that we're observing that NAFLD is forming. And so we treated the ASOs, and we found that, again, this is done in multiple different cell lines, and I'm just showing you too, that there's a huge significant decrease in cell proliferation as well as colony formation and invasion and migration. We're just not showing you this here. And when we tested this to see if the NAFLD foci is significantly lost, we see that when you treat this with ASO I9, for instance, you see there's a significant reduction of these NAFLD foci compared to control. So we think what's happening is that NAFLD causes, when you use the ASO, it causes this exon 10 exclusion, and that causes an out of frame shift that can either be degraded or it can be transcribed into this shorter isoform, but we just don't know if this isoform is becoming a protein or not. We've done some cyclohexamide assays, and we see that there's really no change, but we don't know if it's going through a different mechanism of RNA degradation. And so we're still working on those experiments. And so what we wanted to also do is ask the question as to whether these ASOs are really important for tumor genesis or tumor burden, and if we can use this as a therapeutic target in HCC. And of course, NAFLD is really important for normal physiological functions because it's important for transcription. You can't just inject the ASOs into a human or any organisms and not expect any off-target effects. And so what we opted to do is develop 2 different models. First model is using ultrasound enhanced ultrasound imaging to generate xenografts or mouse models to cause tumor genesis to occur. And so here we have a xenograph model, and this is the needle of us injecting the cells into the liver. And then we asked if over time what happens, and this is IVUS imaging to show that after 8 weeks post-injection of the cells, we can see that there's really nice tumors. But after 2 weeks, when we start seeing little signals, we started treating these mice for an additional 2 weeks, 3 treatments per week, and we found that actually significantly reduced tumor burden in here. One of the problems that we're seeing is that at 25 mg per kg, we're starting to see a lot of toxicity, and that makes sense because NAFLD could potentially be off-targeting affecting, and then what we're using is a pretty high concentration. So right now we're working on using a lower concentration to see if we can get rid of that toxicity and see if we can get a better signal as well. And the other thing that we wanted to do is essentially use these ASOs directly towards the tumor itself. And so we collaborated with Eisenbrae Lab at TJ here, and we're one of the 7 institutions that have a enhanced ultrasound microscopy over here, or imaging, excuse me. And so we took advantage of that by working with him, and their lab has developed these gas field contrast agents known as microbubbles that are negatively charged, and they can be used as carriers to carry our ASOs. They have these in clinical trials right now looking at carrying different chemotherapy drugs for pancreatic cancer as well as liver cancer. And so we said why this is probably the best method for us, mostly because we can't just directly inject NAFLD and expect to see an immediate effect. And so what we did is essentially ask if they can use these microbubbles and then deliver the ASOs for us. And so this is an example of a tumor in a mouse, as you see here. And then these is before we gave them the microbubbles, which is intraperitoneally injected. And so it goes everywhere. So this is, again, the tumor in the same region. And here's the enhanced imaging that you can see. And then when we give it a destructive pulse, you can start seeing that the microbubble starts to oscillate. And then you can start seeing that the microbubbles start to burst. And then after the bursting of the microbubbles, you can start seeing that they slowly come back. This does cause a shearing of the tumors and the cells near it. And so right now we're trying to figure out how to deliver the ASOs as effectively as possible without causing so much shearing. And so far, so our projects are really focused on NAFLD and really understanding how these NAFLD biomolecular condensates can undergo liquid-liquid phase separation to modulate the chromatin, and then really specifically understanding how this modulation can affect downstream mixed signaling. So we're also looking at now downstream of that, seeing how this affects cellular plasticity, looking at different types of phenotypes and genotypes of cellular plasticity. We've also shown here that the SNP is really important, although it's enriched in Southeast Asian patients. They do have patients that the SNP do have a better recurrence rate as well as better outcomes, and that the SNP is promoting exon 10 skipping, and that can then cause the reduction of the mRNA expression. And so far, we've also been able to show that these ASOs are able to target NAFLD and reduce tumor burden. I don't know if we have time, but I just wanted to really briefly talk about the translational stuff that we're doing in pancreatic cancer. It's only 2 or 3 slides. And this is driven by our surgical residents that I work with on a daily basis. And one of them is Saurav Potter, and he's PGY3, and he's in his research year. He's working with us on fatty liver disease in pancreatic cancer. And Brittany, another really talented graduate student, and Kay and Meg, who is starting medical school here at Jefferson. So these are our clinical crew for PDAC, and we call them the PDAC team. And this program is built on the department chair's concept that patients with stage 1 and stage 2 pancreatic cancer can and most likely be eligible for surgical resection, which is considered to be curative. But the problem with that is that insurance is that 70 or 80% of these patients will recur over 2 years. We don't know why they recur. We do know that they do recur at some time and eventually. But what's really interesting is about 20 to 30% of PDAC cases have some type of targetable biomarker. So there's a genetic mutation that could be targetable. But insurance companies don't want to pay for it because they don't think it's necessary. But insurance companies will pay for stage 3, stage 4 molecular profiling. So it's imperative that stage 1 and stage 2, even for that small percentage, to actually get some type of profiling. So we started this program building up from this concept that we should be able to, we want to persuade the insurance company that they should be paying for this molecular targeting or profiling so that we can improve the outcomes of PDAC patients, because 70 to 80% of the time they recur. And so this program called the Jefferson Molecular Profiling of Pancreatic Cancer program, or JAMPAC, program is essentially to strive to generate the data or the evidence to do that. And so our goal is to sequence up to 500 resectable PDAC, stage 1 and stage 2. So these are patients that Dr. Charlie Yeo, our department chair, does, and he does the Whipple. I think he's done more than 1,900 now. And the idea is that we'll be able to use this data to say that there's genetic mutations that we already know that could be used to identify whether this patient should be on a specific targeted therapy so that we can decrease their recurrence rate. And then the idea is that these same patients that come here for surgery will come back if their recurrence does occur, and we would have sequenced them too. So what we've done so far is sequenced 121 PDAC, resectable PDAC, excuse me, stage 1 and stage 2. And of those, about 60% has recurred. And of those 60%, 14 of those patients have had their tumors also sequenced. So this is whole transcriptome and whole exome sequencing. And the idea is that over time, we'll build enough of an N to identify biomarkers or genetic mutations that could be predictive of whether a patient will recur or not, as well as identify mutations that's reoccurring that may potentially help in deciding therapies either before surgery or after surgery. And then we've also done a retrospective analysis of 498 PDAC patients that have been seen here. And the idea is that we would have about 1,000 of those patient cohort, and with the central idea of fatty liver disease as a risk factor for cancer. And more specifically focusing on fatty liver disease as a risk cancer for pancreatic cancer. And we've done some analysis using the Trinetics, and I'm going over time. And what we found was that in 1.1 million patients with fatty liver disease, we found that patients with fatty liver disease are at least 2 times more likely to have a high risk of getting cancers compared to non-fatty liver disease. And these are all age match patients as well. And then when we focus on pancreatic cancer alone, we found that patients with pancreatic cancer actually have a worse outcome when they have fatty liver disease compared to non-fatty liver disease. So currently we're looking at recurrence, progression-free survival. We're also looking at different etiologies as well to really make the mark the point that fatty liver disease is bad for cancer, and this is a novel risk factor for PDAC. And with that, I'd like to thank my team, and I've kind of highlight most of these guys here, and of course all the MDs that I work with, and my collaborators at Brown and Mainz. And I'd like to thank the president for giving me dollars for research, as well as the different foundations that supported our research. I'm so sorry for taking so much time. Thank you.
Thank you so much, Hien. This is really fantastic. I particularly appreciate you're able to actually share many of the unpublished data, especially in terms of the new concept. I felt quite interesting. And I'm going to actually, I'm sure there are a lot of questions in the audience. I'm going to hold off my questions. I'm going to ask you in the, during individual meeting. We're going to probably start from people who have a question in the chat box. And as far as through, I think Sandy is the first person who will ask a question. Maybe Sandy, you can turn on your camera and the audio, and then here, if you can unshare your slides so we can all see each other. And I would like to encourage people that to ask a short question and a short answer. We're probably going to extend maybe another 5 minutes and then finish questions, and then we can wrap up here. Okay, Sandy?
Hi, really nice work. Thank you for sharing that. I have sort of a few interrelated questions. I was wondering what the distribution was of the NELFE was in normal hepatocytes. And along with that, what happens to the chromatin accessibility in the normal hepatocytes or in the tumors if instead of knocking out the NELFE, you use an siRNA just to reduce it, let's say about 30% down to the level of normal hepatocytes. And I suppose along with it, a dose response curve for your antisense oligos on both your hep3B cells, but also on normal hepatocytes to see if there's a therapeutic window.
Right. So these are the things that we're doing. This is part of the grant that we've submitted to. So yeah, so we haven't really measured the expression levels of NELFE in normal cells, but we do know that in at least the transform HHT4 cells that we've been able to work with, they are half the expression level as the hep3B or any other cell lines or cancer cell lines that we observe. But we have not looked at just hepatocytes that you can essentially order from the companies. That was also in the works as well, that we would order these hepatocytes and do the transfection and then see if there's an effect of the chromatin accessibility as well as, I guess, toxicity in these hepatocytes as well. So we don't know the answer to that question, but those are the things that we're doing.
Thank you.
Thank you. Dr. Mishra has a question. Loba, would you like to ask for yourself?
Shin, Lopa had to leave to another meeting, so maybe you can read it.
Okay, so the question from Loba is, does CTCF, one of those complex RNA, the complex for regulating transcription, bind to NELFE or other binding partner for the NELFE that are chromatin modulator?
So CTCF, I don't know if it does specifically, because I haven't really seen in the literature that it does, and we did not see it in our IP. But what we're doing is a bio or an ultra ID assay where we remove the LCS1 and LCS2 and then doing the IP to see if these condensates actually have an effect on, or what is in these condensates and what these proteins are and what they do, and whether these proteins are majority of chromatin modulators. So yeah, we don't know if CTCF is in there.
Okay. The next, I think that maybe this is next question is by Dr. Yi Chen Wang. If Yi Chen, you want to ask question by yourself, I think it may be left the room already. Okay. Perhaps that anyone from the podium wants to audience want to ask a question again. Feel free to turn on your camera. We're gonna entertain one more question.
Hi, I want to ask, when NAFLD regulate MYC, does it bind a specific RNA to does it need to bind a specific RNA, or any RNA is okay to regulate MYC?
Sure. So in our in vitro test tube assay, we used random poly-A RNAs. So it seems like within the in vitro assay, you have to have some type of RNA for it to liquid-liquid phase separation. In the cells, we don't really know. So again, we're doing the ultra ID to kind of figure out what the composition of these condensates are. We're both doing proteomics as well as RNA-seq to identify RNAs, as well as proteins that's within these condensates. In terms of whether it requires to bind to MYC, we found that in liver cancer, when we looked to see if NAFLD binds directly to the MYC transcript, we didn't see that. We found that NAFLD does somewhat indirectly bind to MYC, because we found when we pull down NAFLD, we do see a really slight light band, but it required cross-linking, which tells me that it's a few angstrom away from the actual protein itself, and that's why you're seeing that light band. It's not necessarily a direct interaction, and that's why we think it's a phase-phase separation process. But in terms of whether you need the RNA to bind to MYC, I don't know. I think that Ultra ID assay will really answer that question.
Okay, well, I saw the notes that Dr. Tim Grinton said that there's some technical issue for him, that he couldn't get in the camera works and audio work, so he won't be able to join for the Q&A. Well, again, thank you so much, Hien. This is really a fantastic seminar. I look forward for further interaction. Thank you.
Okay.